Okay, welcome back. So we were just about to describe how new species are named after they're identified. So I don't want you to think of this as some kind of archaic process that doesn't occur anymore. It's very important to know that scientists are still discovering new species of plants, even flowering plants like this flowering tree discovered in Gabon in 2016, Cerdividia solanona. And so new species are being discovered even in the Pacific Northwest, which is pretty amazing. So in order to name a new species, you have to use a process, a formal process. And naming is, the process of naming a new species is called nomenclature. And we use binomial nomenclature for naming a new species. And so I'm going to give you an example of this, Adenostoma fasciculatum. So Adenostoma is the genus name, fasciculatum is the specific epithet, and together the binomial, the two-name system for this species is Adenostoma fasciculatum. That's the species name. Now, after the species name is written, you might see Hook and Arn, or some name here abbreviated, and this abbreviation refers to the author of the species. These are the folks who first discovered and named this species. Now, you'll notice another very important thing here. The genus and the specific epithet are always italicized when you write a Latin name, a Latin binomial name, or a scientific name. You'll also notice that the genus is capitalized and the specific epithet is never capitalized. It's always lowercase. Okay, so those are the rules of botanical nomenclature, of naming plants, that were developed by the International Code of Botanical Nomenclature. And this is a group of taxonomists that meet every year and update the rules, but generally these guidelines are followed all across the world. So all species names follow the same rules. It's not just for plants, it also includes naming algae and fungi. And the goal is to provide one correct name per taxon within a stable classification system. And so in your iNaturalist workshop that you'll be completing at the end of week 3, you'll get a chance to look at some of these online botanical resources where you can discover what the scientific name is of an organism and how to make sure that you have the correct name. And the person that developed this system of naming was Carl Linnaeus. And he came up with this scientific naming system of a two-name system binomial nomenclature. And he developed this to be able to classify organisms easily when they're identified. And he actually wrote a book at the time called, what is the name of that book? It's going to leave me right now. Oh, Species Planetarum. And his goal was to describe every species on Earth. And he actually used more than two binomials, two names for this original system. I think the original system had like 8 different Latin words associated with each species. And his binomial system was just kind of a shorthand for that. So that's what we've ended up using. So the reason why this is important is that these Latin binomials are universal, okay? They're commonly used across the world, regardless of the language, of the native language spoken. And they're important because they prevent the confusion that sometimes common plant names can have, where you might have a common plant that's called one thing in one area of the world and called something different in the other. So these are universal. And again, we have an example, Capsicum annuum. We have capsicum is the genus. Notice it's capitalized. Annuum is the specific epithet. Notice it's never capitalized. And then the whole species name would be Capsicum annuum. Remember, it's always italicized or underlined, very important. And also remember that the authority or the author will often be listed as an initial behind the species, and that's the first person who named it. So the specific epithets are often very descriptive, and the person who names the new species will choose a name that will reflect the morphology of that species. For example, let's say that you have a species that has a winged fruit. You might name the specific epithet Elatus. Let's say that it flowers in the summer. You might name it Estivalis. Let's say that it has bluish gray leaves. You might name it Seaceous. So there's a number of different specific epithets that can be used, and hopefully the authors choose words that would help to describe that species in Latin. So sometimes names change through time, and I'm finding that I'm updating my species lists quite a lot lately, because with the advent of DNA technology, we're getting more accurate understanding of the evolutionary relationships between species, and this leads to name changes. So for example, Andromeda ferruginea was first described by Thomas Walter in 1788, but in 1819, Thomas Natal decided that it was more closely related to plants in the Lyonia genus, and so it was renamed Lyonia ferruginea. And that gets us to classification, the last part of our description, and that's placing objects into some sort of order. So in this classification scheme, we're just looking at the difference between non-vascular and vascular plants, and we have categories of vascular plants where we have seedless, gymno, and angio, and then within angio we have monocot and dicot. So this would be a type of a classification scheme. Species are often fit within rank classification. This is a hierarchical classification scheme, and you've probably heard of some of these. So we have phylum, class, order, family, genus, and species. And what's important to recognize here is that at different levels of, rank classification, you can recognize them because of the ending on the word. So all rankings at the class level and in Opsida, all rankings at the order level and in Ales, and all rankings at the family level, this is probably the most important for you to know, are at the ACE ending, use the ACE ending. And then there's no consistency within the genus or species level endings. So like the rank system, this is part of the Linnaean classification system. Here's like another example of the kingdom, phylum, class, order, family, genus, species. And a word that I'd like you to become familiar with is the word taxon. And this is just a taxonomic group, and it can refer to any of these ranks. So you could describe Acer as a taxon. You could describe Sapindales as a taxa group, for example. And there's a number of mnemonics to remember these designations of domain, kingdom, phylum, class, order, family, genus, species, like do kings play chess on fine grained sand? Do keep putting cheese on 5 green spoons? So anything that can help you with that is useful. Okay, let's take a break and try something. So grab a pen and paper and select a plant species of your choice, maybe looking at the POJAR. And I'd like you to write out the complete rank classification for that. You can look it up on the internet to check your work. So take a few minutes, pause your lecture, and then head back over and we'll finish this up. Okay, so I'm briefly going to run through the major kinds of classification systems that have been used through time, starting with the Linnaean system of phonetic or artificial classification. So phonetic classification seeks to group organisms based on their overall similarity, and usually this is based on their morphology. The pros of a phonetic system is that it helps you to identify things. It aids in identification, and it's using features that you can easily see. So you don't need a lot of expensive equipment in order to just use your eyes to observe differences, for example, in the number of stamens or the position of the stamens, as was done by Linnaeus in his sexual system here. The cons is that you may group species together that appear similar, but that aren't very closely related from an evolutionary standpoint. That's why this system is referred to as artificial, and that's because of something called convergent evolution, where you can have two species that look the same but are very distantly related. Another problem with this system is that it's very subjective. One person might classify like these stamens by their color, and the other one might classify them by their number, for example, right? So it's really subjective to the person who's creating the system. And as I mentioned, this is a problem with phonetics is, for example, with like a cactus from the New World and a euphorbia species from Africa. They appear similar in that they both have modified stems with spines that are adapted for arid environments. Unfortunately, these are not closely related from an evolutionary standpoint. They're actually distantly related organisms that have evolved under similar environmental conditions to develop the same morphological appearance. So that's kind of the problem with the artificial system, but it's a good start. So when Darwin came along with his Origin of Species, this really shed some light on the artificial system and put this in context, because it made people think about the shared common ancestry of species and how all life was interconnected by descent. And so the next phase of taxonomy was really developing an evolutionary framework, and this became called the natural classification system, that all forms of life share a common ancestor. And this shows one of the earliest depictions of an evolutionary tree by Ernst Haeckel. And the similarities and differences in species were seen as a product of evolutionary history. Not only was it useful for identification, but it was thought to reflect the process of evolution. Unfortunately, the taxonomists of this time did not have access to DNA technology to look at the actual sequences and nucleotides in the DNA to be able to look at the similarities at that level, the level that mutation was happening and adaptation was happening. And so this was still somewhat subjective to an individual taxonomist's preferences for what traits he thought or she thought were more ancestral or more derived in their characters. So it was an improved system, but there was still a lot of room for error because of the subjective nature of it. Okay, so here's a little quiz question. The system of plant classification proposed by Carolus Linnaeus was artificial because A, it was based on evolutionary relationships of plants, B, it was based on similarities and differences in floral and other morphological characters, C, it took into account the physiological factors along with the morphological, or D, none of the above. Take a second and think about it. The answer is D. The system was thought to be artificial because it was not based on natural or evolutionary relationships. It wasn't until Darwin came along in the theory of evolution that people started to place these characteristics within this evolutionary context, and that's when we moved into evolutionary taxonomy. So the next phase of classification to develop in time is called phylogenetic classification and cladistics. So like evolutionary taxonomy, it was based on known inferred evolutionary relationships. But now we have DNA as a tool to actually look at the mutations that lead to the classification of different species. So this can get into kind of a semantic argument about what defines a species, right? So there's many different kinds of, there's many different definitions of what a species is. There's like the biological species concept, for example, and we're not going to get into that right now, but just know that this idea of clades or cladistics is a modern method of classifying organisms, which consists of an ancestor and all of its descendants, forming a monophyletic group. So this concept of a monophyletic group is at the core of the definition of what cladistics is. And we're going to discuss that, and I'm going to show you some examples of a clade-based tree in a second. The advantages of this system is that the classification reflects patterns of evolution and that it's not ambiguous. It's very clear cut when you have a definition that each clade or group consists of an ancestor and all of its descendants and forms monophyletic group. If that's the goal of phylogenetic classification, then it becomes very clear how you form a group. And so this is an example of a phylogenetic tree. And when you're reading a phylogenetic tree, the axes here, the y-axis here is time. So the parts of the tree, the nodes, which are representing a common ancestor here, that are further back in time are further back in time. And then the nodes that are higher on the tree are more recent in time. So for example, the tips of the tree here are the taxa. So let's say taxa is, or species C and species D share a common ancestor here. This is the breaking point where their two species diverged. Then you can go back even further in time and find the common ancestor of C, D, E, and F. Or you could go even further back in time and find the common ancestor of all of the taxa in this tree. So the thing to remember here is that taxa which share a common ancestor more recently in time are more closely related than the ones that share a common ancestor further back in time. So for example, C and D are more closely related to each other than C and F, because C and D share a common ancestor more recently in time than C and F do, which is way back here. So now we're going to check our understanding. Which of the 5 marks in the tree above correspond to the most recent common ancestor of a mushroom here and a sponge? So where's the most recent common ancestor of a mushroom and a sponge? Is it B, C, D, E, or A? Okay, let's see. The correct answer is D. The most recent common ancestor of a mushroom and a sponge is D, because that's where the last common ancestor between those two species occur, at this node. So this idea of cladistics is classifying organisms into clades, which consist of the ancestor organism and all of its descendants. So let's practice looking for clades in a tree. One of the ways that you can look for a clade or a monophyletic group in a tree is using the snip test. As you can see in here, if you snip the branch, you get the common ancestor and all 3 of these descendants. Now, that's a monophyletic group, the ancestor and all the descendants. So if we go back here, an ancestor, so if I wanted to make a clade, a monophyletic group of E and F, I would have to circle E and F and this clade, which is its most recent common ancestor. If I wanted to make a clade of D, E, and F, I'd have to circle D, E, F, and this most recent common ancestor. Okay, so the snip test. Now, it's not a clade. Let's say that you thought that these 3 species were going to form a clade. This doesn't pass the one snip test because you'd have to include the common ancestor of this, this, this. So the most recent common ancestor of these 3 clades is all the way down here, right? So that's not forming a monophyletic group because you have to do 2 snips, okay? Now, paraphyletic group is when you have the common ancestor, but not all of its descendants. So this is also not a monophyletic group because it leaves out all of these descendants of this common ancestor, for example. So here's another picture to help you understand the difference between monophyletic, paraphyletic, and polyphyletic. So here's a partial evolutionary tree for vertebrates. And you can see that the yellow is including, it looks like reptiles and birds. And you can see that the yellow includes the common ancestor of reptiles and all the descendants, right? Now, the paraphyletic group contains the common ancestor, but it leaves birds out. And that's understandable if you think because reptiles are different than birds, but in an evolutionary construct, they are descended from this ancestor, so they should be included in this clade. So that's paraphily. And then polyphily is saying, okay, reptiles are cold-blooded, but birds and mammals are warm-blooded. So we want to group mammals and birds together. However, this doesn't include, this is an unnatural grouping because it doesn't include the common ancestor of mammals and birds. And so it's a polyphyly. It's unnatural to group them that way because it doesn't include the common ancestor and all the descendants of those two taxa. So that's kind of some information about cladistics. Now let's practice to see if we can find a monophyletic group. So among the following diagrams, which taxon represented by the shaded region represents a monophyletic group? Is this a monophyletic group? Is this a monophyletic group? Is this a monophyletic group? So which one is a monophyletic group, or is there more than one? So take a second and consider which answer you'd like to choose. All right, the correct answer is D, taxon A only. This is the only one that has the common ancestor and all of its descendants in the shaded region. This one has a common ancestor, but just these descendants and not including those. So that would be a paraphyletic group. Whereas C represents all of these descendants, but doesn't include their common ancestor. So that's a polyphyletic group. So that's an introduction to the classification systematics of plants, a little bit about tree reading. Hope you enjoyed it. We will now stop, and we look forward to talking about this more in class. See you soon.